Got it. And yeah, and there's 3 chapters on aging. If you're interested in aging, why it happens, how, and it's all written for physicists, biologists. The math is friendly. And today I'm going to talk about chapter 5, fibrosis. All right, so I'm going to share screen. I am sharing screen, right, Paula? Yeah. All right. So this is going to
Yes.
be a whiteboard talk,
Yeah.
and you see Sydney Lamb said, "Hi, I am a doctor," on the chat. I hope you can see the chat. Oh, and Amro doesn't have a functional camera. That's fine. Hi, you can turn off your camera now. You don't. I just wanted to say hi. You don't have to be with your camera open, of course. Hi, Sydney. So I want people to chat questions here. Okay, chat your questions here. Okay, so I'm gonna talk about fibrosis. Fibrosis is excessive scarring, and it's a problem that cuts across medicine. So you have, especially with age, but not necessarily, organs become a scar and they lose function. You can have liver cirrhosis, which is liver turns to a scar. You can have lung fibrosis, and in the end, you have to replace the lung or the liver. There's no cure. Progressive diseases. You can have after a heart attack, the cells that die because they don't get oxygen become a scar tissue, and that's good. Scarring is good if you don't want a hole into the tissue, right? So it's very important. Or when you have a virus like hepatitis C, you encapsulate it. If you can't get rid of it, a piece of something stuck in you. But then fibrosis is a pathology that is bad for you. So it's probably responsible for or involved in 50% of deaths. Fibrosis also enhances the risk of cancer. Like there's no liver cancer without liver fibrosis almost. How are your nephrology training? Oh, Itzhaki asked Sydney. So Sydney, if I say something that's inaccurate, please tell me. Okay, and also all the doctors here, anyone. And so we want to understand fibrosis. There's no cure. It's very extensive, and we want to think about it. It's also very complex, and because it's so complex, it's perfect for physicists to try to find an angle of simplicity. And there's a sequence across medicine, which is injury. So the tissue is injured. That leads to inflammation. So redness, swelling, heat, and pain. And that can have two outcomes, two different outcomes. Basically, you can have healing if the tissue can regenerate. Like the skin, right? You get a little scar on your skin, it goes away after a couple of weeks, healing. Or it can go to fibrosis. So we have one system, two outcomes. So our first question is, in this talk, is how can one system, wound healing, have two outcomes? And we're going to, of course, understand how this multi-stability, two or more outcomes, is important for understanding fundamental properties of fibrosis. For example, inflammation. Doctors try to stop inflammation, and sometimes very quickly, like 1 day, 2 days, because if it goes beyond a golden period, you're bound to have fibrosis, even if you stop the inflammation later. So where is the golden window of about 2 days come from? Where does the golden? Okay, and of course, how can we reverse or prevent fibrosis? So in this talk, we'll go from some experiments to a mathematical model, understand what are the targets, and then actually get all the way down to molecules in mice. So we're going to show you a couple of you say preclinical drugs in mice that can reverse fibrosis in heart attacks and in liver cirrhosis. So we'll go from math to targets. And also, my goal is everyone will understand everything in this talk. And if you don't understand, it's my responsibility. So please write down the question. Okay. I'm going to stop now, actually, until somebody writes a question. I'm serious. I'm not going to continue until somebody asks me a question by chat, just to kick things off. Who's going to be brave? Matthew Smart. Is 2 outcomes too limiting? There'll actually be multiple outcomes. Let's wait for another question. Who's going to be brave and ask me a question? Joseph, what fraction ends in healing versus fibrosis? Depends on the organ. It's a very important question. The deeper the injury, or so deep injuries, large injuries, repetitive injuries, more chance for fibrosis. The less regenerative the organ, the more chance for fibrosis. In young people, more chance for healing. In old people, more chance for fibrosis. Chance for fibrosis raises with age. I think the risk of progressive fibrotic diseases rises exponentially with age and drops at very old ages because everybody who could have gotten sick has already gotten sick. What is the starting point really the same? Yeah, I would say that the starting point also makes a big difference, as I'll show you. Vitali, how many cell types should we consider? So there's many complicated cell types, and as I'll show you, as a physicist, we're going to focus on the 2 that I think are essential without trying to belittle or to devalue the work on other cell types, which I think is just having 2 cell types is enough to capture the essentials. What makes study fibrosis and reversibility so difficult? Sydney asks, Dr. Sydney Lamb. And what do you think, Sydney? Can you type it? And also, I'd like Matthew, Joseph, Shrini, and Vitali to say thank you if I answered your question. And if not, just say what I should answer. Okay, I'm so controlling. It's because I want contact with you. I want to learn from you because you're so many people in the audience, so educated, and I want to learn something new, not only tell you. Matthew, I'm waiting for you to say yes, you answered my question, or oh yeah, you did. Oh, there's more questions. Are there tissues don't undergo fibrosis? Yeah, in the embryo, the fetus, you make a damage, it doesn't undergo fibrosis. The heart of a 7-day-old mouse regenerates, but an 8-day-old mouse fibrosis if you do a heart attack. And salamanders regenerate. I mean, there's no fibrosis. There's senescence, scarring, regeneration. So that's very plastic in the animal kingdom and across our ages. Joseph said, okay, great. Why would fibrosis lead to cancer? So cancer, we used to think of it as only mutations, but now we know that it's an AND gate between mutations and a permissive environment. And that permissive environment is often inflammation and fibrosis. Some cancers are really inflammation-related cancers. The stiff matrix of the scar helps cells do metaplasia, which helps cancer. And the inflammatory environment sets up a situation where cells don't want to grow, and then the mutant cells have a growth advantage, and many other things. So cancer is an AND gate over the environment and mutations. Okay, I'm gonna stop with the questions right now. I so love your questions. And remember to say thank you, or you answered me if I answered you. Can fibrosis result from any prolonged inflammation? No, there's different kinds of inflammation. There's chronic inflammation of many different kinds. But the inflammation that happens after injury, I'm going to talk about. And it can result from that, but maybe, as I said, in the fetus, it doesn't. Vitaly, well, nice to hear your proposal first, then more to discuss. You're right. Let's go for it. Okay, so here's a primer about what is wound healing. And so I'm going to draw for you here a typical tissue. These are the epithelial cells of a tissue. Here are the nuclei, yeah. And I'm also, for you, I'm going to draw a blood vessel here. It's a blood vessel. And I'm going to open a window into this blood vessel. And here is a red blood cell. Okay, that's our tissue. And now there's some damage, some injury to the tissue. This cell dies. This cell gets stressed. So when cells get injured, a lot of times they become senescent cells, which means they stop dividing and secrete pro-inflammatory factors after a few days. Senescent cells. And the damaged cells very quickly secrete all kinds of factors that do two things. One is they cause monocytes in the blood to go through the blood vessel and become macrophages. So this will be one cell type that we'll be interested in. We'll call it M, macrophages, which, of course, there's neutrophils in the first day, gobble up bacteria. But I'm going to talk about the scale of days, weeks, months. And so we're going to ignore a lot of things. One is because of time scale, I'm going to ignore neutrophils. There's also endothelial cells and mast cells. A lot of things I'm going to ignore here. I'm going to focus on macrophages. And the other important cell type is fibroblasts, fiber-forming cells, which are tissue-resident fibroblasts. And signals like TGF-beta make them become large myofibroblasts. We'll call them F also because so myofibroblasts. Myo means muscle, and myofibroblasts are filled with fiber, with muscle, actin, and motors, and because they can generate force and contract the wound. And second, they secrete the fibers, all kind of stiff collagens and other matrix components that make up the scar. And the fibroblasts and macrophages talk to each other with secreted cytokines, chemokines, growth factors. And they are kind of considered to be important cells. And of course, endothelial cells, if it's vascularization, the epithelial cells play a role. But I'm going to focus on fibroblasts and macrophages. Sydney says, fibrosis is complicated, complicated, involves cell signaling, death, and subsequent remodeling, epigenetics, too complex to study in short. So I'm going to try to find an angle of simplicity, but maybe it is too complicated. What's the role of fibroblasts in fibrosis? Does killing help? Killing fibroblasts help? So we'll see. Fibroblasts, of course, make the fibers, but these are myofibroblasts, right? So you don't want to kill every fibroblast because the tissue-resident fibroblasts are very important. Now, I'm going to tell you, our work started with an experiment by Uri Alon, Shu, Ruslan Medjitov, and me, published in Cell 2018, where we want to ask about the circuit of fibroblasts and macrophages, and the approach was in vitro. So you take a plate, and in this plate you put some fibroblasts and some macrophages at different concentrations, let's say 3-to-1 in this case, let's imagine. And in another plate, you do a different concentration of seeding, let's say more macrophages in this plate. And then you measure the dynamics, the divisions, over, let's say, 4 days in each situation. And you plot the plots. It's going to be important for us in this talk. Where on the x-axis is how many fibroblasts are in the plate, and on the y-axis how many macrophages are in the plate. Now, of course, macrophages come in a continuum of states. It could be M1, M2, pro-inflammatory, anti-inflammatory. Myofibroblasts are of a continuum of states. We're just going to count cells here again, another simplification. And the beautiful thing about this experiment is that it gives you the phase portrait of the system in one fell swoop. How does this work? Suppose I started with this plate that had 3 and 1. So at 3 and 1, this is the beginning. And after 4 days, it went like this to this point. That gives me a dynamical arrow. And I do that for different initial conditions, right? Different initial conditions. And each initial condition gives me another arrow. So macrophages on their own, here on the y-axis, they crash. And fibroblasts also crash to 0 unless they're larger than a critical concentration in which they start growing. And at very high levels, they shrink. So in short, understand basically the differential equations that govern the system from this experiment. That was the point of this paper. And you can determine that there is a steady state fixed point, a stable fixed point here. So the cells flow to this fixed point. And we called this, we made up a name for this, hot fibrosis. Why? Because fibroblasts and macrophages together. Hot from cancer means that there are immune cells there, like T cells and B cells. Here we borrowed that word. Hot fibrosis means fibroblasts plus macrophages. It's a new word for pathology. By the way, I forgot to say, when there's healing, I'm now looking at this fibroblast and macrophage side of the plot here. I'm here now. When there's healing, the fibroblasts, the myofibroblasts, and the monocyte-derived macrophages, they all die in apoptose. So there's the scar gets digested by metalloproteases secreted by the macrophages and by the tissue, and the senescent cells are removed by NK cells and macrophages. The epithelium regrows. And like a kid, you had a scar, 2 weeks later, nothing. Fibrosis, the cells stick around. That's why this point here, where fibroblasts and macrophages vanish, so macrophages crash, fibroblasts crash, or if there's too few cells, they crash. We call this, we're going to call this the healing state. It's like a mimic of the healing state because those cells, fibrotic cells, are gone. By the way, when you put fibroblasts and macrophages on a plate, the fibroblasts become myofibroblasts very quickly. And the macrophages are bone marrow, they're like monocyte-derived macrophages. So these are like similar cells to what you find in fibrosis. And here's another, but then we discovered there's another point here. It's a semi-stable point where fibroblasts can support themselves. They don't need, there's zero macrophages, right? So, and we call this cold fibrosis. And there's a special and important line that separates the situations that lead to hot fibrosis and cold fibrosis. It's going to be very important for us for preventing fibrosis. It's called the separatrix. It's a dynamical system term for the line that separates the basin of attraction, which leads to healing, and the basin of attraction that leads to hot fibrosis. By the way, these equations immediately allow you to write down the circuit, as I said. So fibroblasts support macrophages with growth factors, like CSF1, we know from the in vitro experiment. Macrophages support fibroblasts by other growth factors like PDGF-beta that we know from experiments. And importantly, fibroblasts can also support themselves with PDGF-alpha and other growth factors in an autocrine loop. And that's essential to have that fixed point down on the x-axis that we call cold fibrosis. Fibroblasts support themselves. And are you guys with me or against me? With me. All right. How do I go back to? Oh yeah, this. All right. And so we know the circuit here. And I just want to say that if you tweak this circuit parameters, for example, if you make the macrophage to fibroblast arrow a little weaker, you can have a situation where the cold fibrosis state becomes stable and has its own basin of attraction on the computer. So tissues can have cold fibrosis or hot fibrosis. I'm going to talk which tissue has what, because it turns out that this is very interesting and important distinction. All right, so it depends on the circuit if you have cold fibrosis stable or not stable. It depends on the tissue, etc. What's the role of fibrosis? Is cold fibrosis a saddle point? It's a saddle point unless the circuit is such that it's stable. And is treating cold fibrosis easier than, I'm going to talk about how to treat. Cold fibrosis is easier to treat. How is the equation for the separatrix derived? Amruta. And say yes if I answered your question, Sylvia, okay? Because I might not answer your question correctly and I want to know. So the separatrix is derived, it's the stable manifold of this unstable fixed point. So trajectories on the separatrix flow. This is like a mathematical thing. They flow to the unstable fixed point and then away. That's how it's determined. And that's in general in dynamical systems. How is the fibroblast state altered by hot cold fibrosis? Yeah, fibroblast state, we also measured experimentally, and the states are indeed very pro making a lot of collagen fibers, stuff like that. Proliferation, inhibition of metalloproteases. So there's a whole program. But now we can be simple. All right, great questions. I'm going to continue. Sonali, I'm going to answer your question in a sec. Okay, now I'm going to simulate for you on the computer a wound. Okay, so let's start with a small injury. So we have some tissue-resident fibroblasts and macrophages, and there's an influx. The first thing that happens in injury, as I said, after in a couple of days, a big influx of these macrophages. And let's say that influx ends here because it's not such a big injury. Now the macrophages cause the fibroblasts to divide, which causes the macrophages to divide, but it's not enough. Here, actually, there's another unstable fixed point here. Amruta, this is determined by the crossing of two null lines, this unstable fixed point, which makes things very slow here. So actually dynamics here slows down. That's why it takes a couple of weeks for the injury to heal, even though cells turn over in a day or so. But you go back to healing. So this is what healing looks like. And indeed, when we look with Shalevitzkovitz at a mouse liver that you injure, and you get some injury, after a couple of weeks it heals, and you count macrophages and fibroblasts, and you get this kind of loop. Amruta says, got it. Now let's do a longer injury. So let's say now for 3 days, we have macrophages and inflammation. Now this is our initial condition. Macrophages make fibroblasts divide, but it's not enough. We crash. We slow down. But then we go here. Why? Because we're in the basin of attraction of the hot fibrosis fixed point. That's how a tiny difference in initial conditions can lead to a vast difference of healing versus hot fibrosis. And that's how one system can have two outcomes by stability or multi-stability. And it does depend on the initial conditions. This also explains why we have this golden window of 2 days, because of the by stability of the separatrix. And now we come to our third question. How can we reverse or prevent fibrosis? Sonali asked, is there a direct path, circuit, trajectory from hot fibrosis to healing, or is it always versus cold fibrosis? So let's talk about that now, because I want to reverse hot fibrosis, cold fibrosis, etc. Basically, when an organ gets to hot fibrosis, it stays there. That's the common wisdom, at least. Even though it's beginning to be doubted, because, for example, once there were good drugs for hepatitis C, which makes fibrosis, that gets rid of the virus. Some doctors bothered to look a year later at the patients after they were well. They did the ultrasound, and the fibrosis was gone, which means that it's actually reversible to a degree, probably because of the metalloproteases secreted by the tissues, etc. So, how can we revert fibrosis? So on the computer, what we can do now is play with each arrow. That is to say, change the parameters on each arrow, the death rates, the division rates, and see which ones increase the basin of attraction. By the way, can you see my cursor move like a little pencil? Can somebody tell me?
Yes.
Like a movie is now. You can. Okay. So this is the basin of attraction of the what increase the basin of attraction of the healing state. So we found that the parameter that does this the best is this autocrine growth factors. If you want to inhibit the autocrine growth factors. Inhibit the autocrine growth factors. Why? Because when you inhibit the autocrine growth factors, Let's look at the x-axis. The weaker the ability of fibroblasts to maintain themselves, the more you need in order not to crash. So this unstable fixed points moves here when you inhibit the autocrine loop. And it drags the separatrix with it, actually. So now we've enlarged the area, the range of situations, which goes back to healing. So that, we now this injury will heal. Sorry about the messy. Did I explain myself? And if I push more on the autocrine loop, there's going to become a critical point where these two fixed points collide. And then they both vanish in what's called a blue sky bifurcation. And now the separatrix looks like this. And there's no cold fibrosis anymore. And a lot of situations go to healing. So if I have cold fibrosis, this is enough. If I have hot fibrosis, I need a double step. So it is harder. Somebody asked me. I need to first destroy some macrophages, which is easy, like dexamethasone or whatever, and then get into the basin of attraction of the healing state and flow back here. What cell composition governs the separatrix? Sonali, I don't know how to answer that, but I just answered your direct path from hot fibrosis to cold fibrosis. Itzhaki, does zero in the phase portrait mean a basal level of fibrous macrophages? I guess there's always some yeah, zero here means resident macrophages and fibroblasts, you can say. And that's actually what we measure, tissue resident, because every tissue has fibroblast macrophages, blood cells, and the special cell, like violinists, like whatever it is, liver cells, skin cells, that do the actual work, but they're very, very specialized. Like a concert violinist, if their toilet or like their sink gets clogged, they call the macrophage to bring in the repair person. The macrophage is like the butler, and the fibroblast, like it makes the fibers. It's like the carpenter. See? So okay, so we have our formula for we're doing great on time. So I would say that inhibiting the autocrine loop is predicted to prevent and reverse fibrosis. And I want to say also that you don't have to push it all the way to pedal to the metal. The nice thing about this blue sky bifurcation, the annihilation of two fixed points, it happens at a modest reduction of the autocrine loop. You just need, it's like COVID, you don't need to put the replication number to 0, only below 1. You just need each fibroblast to make less than one new fibroblast, you can say. And that's very hopeful because these growth factor loops are actually made of many secreted factors. So maybe if you inhibit one just enough, it will be enough. And that's why when experimental collaborators heard this talk a few years ago, they said, I want to do experiments on my system, and I want to tell you what they found. Okay. Adriana, you can also ask a question. Listen. I'm going to tell you now 2 experiments we did, one on heart and one on liver, in collaboration with 2 groups. So I'm going to divide the screen here to the heart and the liver. Okay, heart and liver. Brian, what role does stochasticity play in the system? I haven't considered that too much, even though in my book, aging, stochasticity plays a major role. That's why identical twins, same genes, same environment, identical twin mice, people die at different times, like 2 unrelated people almost because of stochasticity. In this case, I would guess that if it's a small injury and we're talking about small numbers of cells, something like that, it maybe makes a huge difference. But we didn't find a need to include stochasticity for what I'm talking about. I hope I answered your question, Brian. And if you have a reason for that question, please write it in the chat. Why do you ask that question? And Sonali, you write to me why you asked that question about the composition, because I didn't get it, so I want to understand it. How compare the actual parameters measured in vitro to the 3D in vivo situation? So I would say that in vitro, it's very different. The growth factors are different. That's why we did experiments on in vivo, and the growth factors are different stuff. But I think the principle bistability, separatrix, inhibiting the autocrine loop is the same. So that's the role of in vitro and math, in silico, back of the envelope, all these things, and just to give you the concepts. But I'm gonna now let's get in vivo, guys. Okay, let's get in vivo. Let's get in vivo. Let's get in vivo. Let's get into mice. Heart attacks or myocardial infarctions. So heart attack will be a very big killer, and myocardial infarctions. Okay, I'm really bad at spelling. Hardy attacks. Hardy. MI. And this is a collaboration with a joint PhD student, Shoval Miara, working with Eldad Sacho, the lab next door, who is heart regeneration expert. And this is now, you can find this on bioRxiv 2023. It's under review in molecular cell. Okay. So what did Shoval do? Vitali, which cell? You guys are so generous with your questions. Yeah, Abi, it can be multi-stable. So in certain situations, you can have both cold fibrosis and hot fibrosis, indeed. Sonali, I want to know quantitative ratios of macrophage fibrosis comprising separatrix. Yeah, there is a quantitative ratio, exactly, of fibroblasts and macrophages. So that's why depleting macrophages can have very different effects. If you're below the separatrix, it's good to deplete macrophages. If you're above the separatrix, above this point here, it's bad because you can go from hot fibrosis to cold fibrosis, which is probably worse because macrophages soften up the fibroblasts with the Mertal proteases, and you don't want to go to cold fibrosis because that's stiffer. And so it's very important. I agree with you, Sonali. All right, so Shoval did the following. He looked at, when you do a heart attack to mice, which he does, you tie up a little blood vessel that gives oxygen to the heart with a little string that's called ligating it, and then the heart cells don't have oxygen and they die, and then there's massive senescence and fibrosis because you don't want the heart to have a hole in it. That's critical. So there's fibrosis. As I said, 7-day-old mice or below regenerate, 8-day-old and above don't regenerate. So if you take a mouse heart and do a cross section, and you do that, you get an area of fibrosis. This is a serious red staining, right, for fibrosis. You get this kind of scar. And in fact, you have macrophages and fibroblasts in the beginning. They peak in 4 days. Then the macrophages are found only in the layer between the healthy and dead heart, and that layer shrinks. And what he discovered is it's cold fibrosis, actually. The heart has cold fibrosis. That's to say, after 30 days, there's no macrophages in there. Fibroblasts that support themselves, a very fibrotic state, etc. And I want to say that other tissues have hot fibrosis. We think that congenital heart, like hypertensive heart, if the heart works too much, there's a heart failure. That's not a heart attack, but that we think is hot fibrosis. Also, digital pathologists in Germany, Feuerhacke et al., showed that and used these concepts of hot and cold fibrosis, which we are thrilled are becoming important for pathologists, to show that in kidney, glomerular fibrosis is hot fibrosis, and interstitial fibrosis around the tubules is cold fibrosis, and it's affected by fields of hypoxia and inflammation. And as I said, there should be different treatments for them, and I'm really excited to see where it will go. Okay. In vivo, why is cold fibrosis not resolved by new macrophages? When there's cold fibrosis, there's actually a basin of attraction to cold fibrosis, or sometimes, depending on the circuit, there's even no hot fibrosis state. So the macrophages are not going to help in cold fibrosis. Vitaly, I hope I explained to you. Just the parameters of the circuit determine which fixed points are stable. Can we tempt the fibroblasts to be dependent on macrophages again? Wow, what a question. Look, so what Choval did is he did single-cell RNA-seq of mouse myocardial infarction and human myocardial infarction, and he did these T-SNE plots, right? So you get the myocardiocytes, and you get all the cell types, and you get these cell types, which are, so you can find, for example, these will be here the resident fibroblasts, and these here will be the myofibroblasts, the bad fibroblasts. They appear only in the diseased state. So these myofibroblasts are RF for us. These are tissue resident here. Okay. And then he asked, he looked at these fibroblast cells, myofibroblast cells, and he asked, which RNAs do they make? This is single RNA seq that creates a secreted molecule that's a growth factor, and they also make the receptor for that growth factor. In other words, he screened for autocrine myofibroblast growth factors, but importantly, that are not sensed by other cells. They're specific to myofibroblasts because you don't want a drug that'll, like in fibrosis, people have tried the major players like TGF beta, FGF, blah blah F, PDGF, and they're bad drugs because they're so pleiotropic. What we want is something that's specific to the disease state. We don't want to mess up the rest of the body. And it turns out that in the list of humans and mice, there's a list of growth factors, and he discounted things that haven't worked before, and he looked for things which are druggable, and he found, he focused on, and this is a fantastic thing, he focused on tissue inhibitor of metalloprotease 1, which is a protein that has a well-known function in fibrosis, which is inhibiting the metalloproteases that cut up the collagen and the ECM, but TIMP1, 2, 3, 4, TIMP1 has the least of this inhibitory activity, and it turns out, and Cheval proved this, it's actually a myofibroblast growth factor, a cardiac myofibroblast growth factor. But in vitro, he put some recombinant TIMP1 and they grew, and then he put on some antibody that neutralized TIMP1 and they shrank. So it's a cardiac growth factor. And then he did the important experiment. He took the mice, and he gave them heart attacks. I don't like the red color. I'm switching back to black. And then he injected them with antibodies against TIMP1 or with control antibodies. So these are neutralizing antibodies against TIMP1, right? At the heart attack, and 3 days later, we don't have experiments for the developed heart fibrosis, only for the beginning, but we'll have them in the liver. So, and what he found was that antibodies against TIMP1 reduced fibrosis by 50% and made it not go through the heart. So the mice were in much better shape. Inhibiting the autocrine growth factor reduces fibrosis. And the heart doesn't heal, it regenerates, but apparently those cardiac muscle cells can pull themselves together, and you have a healthy heart basically there. Okay. Any questions about that? Sydney, what makes glomerular fibrosis different from interstitial fibrosis? I really don't know. I would guess that the fibroblasts that over there are wired so that they go to hot fibrosis, and the ones near the interstitium is wired to go to cold fibrosis. And also it could be due to hypoxia. The more hypoxic situation might go to cold fibrosis. So these kind of things are at the forefront of research in our little field. Wow, so many questions. What should I do? No, actually, Sydney's the last question. Okay, great. So so this is the next story. The final story is in vivo mouse NASH liver. So NASH, black. This is liver cirrhosis, which is fibrosis of the liver. NASH is a condition that is very common. I think 30% of the world population has fatty liver. A certain fraction of those go to NASH. A certain fraction of those get cirrhosis, which means the liver becomes like a piece of tire. And then you need a liver transplant because the liver does a lot of important things. And a few percent of those people get liver cancer, which is a deadly cancer because you discover it when it's too late. So it's a very fast rising disease and very important disease, and again has no clinically approved drugs. And so we worked here with a world expert on NASH cirrhosis. This is Friedman Lab at Mount Sinai. Sammy Wang is the first author in this paper in Science Translation Medicine 2023. The Friedman Lab developed the NASH mouse, which is a good model system for NASH. You feed the mice with a liver toxin, CCL4, and high-fat diet for 24 weeks, which makes their liver extremely fibrotic, as you can see in this serious red stain here, after 24 weeks. They did exactly what Choval did, single-cell RNA sequencing of human and mouse NASH and controls. And again, they found that the fibroblasts came in two flavors. There's hepatic stellate cells, which are the fibroblasts of this one is the tissue resident fibroblast. And then only in the diseased livers, you have HSC2, which are a different cluster, which are the R myofibroblasts. And then they looked at HSC2 and asked, using NicheNet and all these kind of things, what is their autocrine growth factor? And again, they got a list. They discounted the usual suspects, things that don't work, looked for druggable targets, and they came up with the following picture, that those myofibroblasts in the liver, they look a little bit like neurons. They have these processes. They touch each other. And they secrete something and they receive it. And that's something that they focused on is neurotrophic factor 3, which is a neuron growth factor, but also a stellate cell, HSC2 growth factor, and its receptor, NTRK3, which is a famous cancer tyrosine kinase receptor. Were the mice happy about the high diet? I think, yeah, mice are mammals and they have emotions, right? They have a limbic center. I don't think they're happy about the CCL4 liver toxin. I don't know if they're happy to be alive, I have to say. It's a good question. Why do you ask, Itzhaki? And of course, we're doing this to help hopefully cure people or in one day or contribute to the discussion. So NTRK3 is a tyrosine receptor kinase, and there exists a drug that's a tyrosine that was like it's a failed cancer drug that is a tyrosine kinase inhibitor. And what they did is they took the mice, gave them the 24 weeks of this torture. So now they have the really bad fibrosis here. And now this is irreversible. Right? You stop the treatment, the fibrosis is not going away. And then over 4 weeks, they injected this drug that you can find in the paper, of course. And they checked the livers. And again, there was a reduction of 50%. of fibrosis and regeneration of big patches of healthy liver. Generation of 50% healthy liver. Inhibiting the autocrine loop reversed this fibrosis. So here we have in the heart, it's an acute situation. In the NASH liver, it's a chronic situation. And I think this is the end of my story. Now, I left a lot of time, 14 minutes, for you to ask me questions. Because I want to, or tell me about a fibrotic disease that I don't know about, because I think this might be general. Because as Michael says, it's two universality. We have two diseases here. Okay, Philip Schaefer, was the regenerated liver functional? Yeah, it was functional, and also apparently fewer cancers formed in this mouse. We were checking that out. We don't know that for sure. Any side effects of the autocrine inhibitors? So in both cases, we couldn't see any side effects of the autocrine inhibitors, and we think that is because they're only made by inflammatory, like myofibroblasts, basically. So I guess I wouldn't take them after you had heart surgery or surgery, because you want healing to progress well. But I don't think there was no detectable side effects anyway. Multi, is the 50% stochastic sum of smaller pieces, or are we missing another big component? So what you're asking is, why only 50%, right? Why only 50% reduction? I don't know. It's a really good question. Of course, could be another big piece to the puzzle. David, what about TB granulomata? What about where fibrosis walls off foci of TB infection? So David is telling me about something I don't know about. TB infection has fibrosis that protects the bacteria, I assume. Am I right, David? If yes, can you write a little more information about that? I would appreciate it if you do that in the chat. Matthew Smart, by the way, I'm seeing some people ask many times, which is good, but I want those of you that are normally don't ask questions to dare to ask me a question, because I'm sure you have either something very important to teach me or some clarification that many other people will benefit from. So this is your chance, because I'm very friendly and I respect your question, I promise you. Which organs, e.g. mammals, do you expect the proposed model to apply to? What example of an experimental measurement in post-injury inflammatory tissue that would indicate this simple model needs to be reconsidered or modified? So Feuerhake group already extended the model by looking at the kidney and seeing that cold or hot fibrosis could depend on fields of inflammatory cytokines and hypoxia, which they measured in a brilliant way. So that affects the parameters of the same circuit and can make a bifurcation. So what cold fibrosis stabilizes hot fibrosis. So that's a really interesting topic. And of course, other organs like lung, skin, where you have hot fibrosis is what's called keloids, where you have a very hot kind of fibroblastic macrophage tissue. And cold fibrosis are these hyperproliferative scars, which are just the regular scars, or two different kinds of scars recognized by dermatologists. And I think they can fit this cold and hot fibrosis scheme. I'd love to know. And there's fibrosis in bone marrow. And teach me about more fibrosis. If you guys know about fibrosis, ask me specifically. Do uncontrolled autocrine loops direct fibrosis to liver cancer? Wow, what a question. So the fibrosis in the fibroblast, the liver cancer in the hepatocytes, I don't know the answer, but I would guess that what the hepatocytes are feeling are a lot of inflammation and a lot of stiff matrix. Cells that are on a stiff matrix do what's called metaplasia. They change to other kinds of cell types because of the damage. And when that happens, that's very vulnerable because of epigenetic changes, and then they can become dysplastic and then cancerous. So there's something about the microenvironment that changes that is being studied now. It's pro-cancerogenic. And Srini, did the inhibitor treatment change the cell composition of the fibrotic liver other than fibroblasts? I don't know. Philip, if you would extend this model to fibrosis, which cell type component would you add first? In the paper, we actually added the epithelium or the normal tissue regeneration to it. I would now also add the vasculature, which plays a signaling role. I guess those are the two I would start. Matilda, do you think it could be different in implant fibrosis, considering that some input might remain since the implant remains? Oh, I never thought about that, but the implant fibrosis, so the implant keeps doing the injury, right? Maybe if we coat the implant by autocrine loop inhibitor, we stand a chance to make it go away from fibrosis. Maybe the fibrosis there is actually good for you. Could be. So I'm not sure. Itzhaki, does aging affect the process of fibrosis? Oh, does it? Oh, it's a superb question. So here we have our tired old face portrait. I'm going to use it again and remind you of our original separatrix. Let's make it bold. Oh, can I? Brush. Let's go wild. It's not so wild. With aging, Miri Adler in her paper, by the way, there's a theoretical paper that if you guys like theory, Adler, iScience 2020 on this fibrosis model with all these separatrix and stuff. What she showed is she analyzed many things that happen in aging. Cell division slows down, inflammation markers go up, blah, blah, blah. And all of them do this. All of them push the separatrix in the bad direction. So now an injury that would have healed when you're old, let's say when you're blue, right? You're old, blue. Same injury is now in the basin of attraction of fibrosis. And that's one reason, perhaps, where you get fibrotic diseases exponentially rise with age. By the way, there's a paper about why it's exponential rise with age that could be interesting for you. Aging 2021, why exponential rise with age of hundreds of age-related diseases. We study aging, so we have this whole picture about aging and age-related diseases, which is in the book, chapter 6, 7, 8, and also on our website. You can find me. You don't need to buy the book, by the way, because I have all the courses where you talk about the book on the website. You can find out our theory on aging and can explain things like IPF, why idiopathic pulmonary fibrosis, a disease that rises exponentially with age. Luckily, only in one in about 100,000 people, but progressively your lung turns to scar and you unfortunately die within a few years because you can't breathe. So what's going on there? We can explain. Is not whoa. Joseph H. Nasser, is there any genetic evidence for this model? People who never get fibrosis have mutations in autocrine growth factors. That's a great question. I don't know. Like people with keloid scars or people, maybe you'll find variants in TIMP1 or something like that. It probably is known. I just don't know it. Marco, are there animals where fibrosis is more or less prevalent? Could this highlight some important extra players? Yeah, for sure. So naked mole rat and things like this, less fibrotic. Mice, young animals more than old animals. Regenerating animals like lizard tail and stuff like that, different organs. So comparative biology of this would be fascinating because maybe you can rationalize what are the different parameters in their model, in their it's great questions. David, can you tell me about TB granulomata, please? Leonie, what do you think is the mechanism behind the separatrix moving with increasing age? So, in fact, there's many mechanisms, but I think that the way I look at it now, mammalian aging is driven by damage in stem cells that causes damage to differentiated cells that become senescent cells. Senescent cells, you heard about, senescent cells are what happens in normal healing. They're very important when you're young. because they stop the cell from regenerating and call in the immune system to remove them in an organized way. But when you're old, you have more and more problems with your stem cells. So you have more and more micro injuries generating senescent cells. And then the cells that remove senescent cells, which are NK cells and macrophages, the garbage trucks of the body become overloaded with their task, and they can't do the normal task of removing cancer cells and infection. And then senescent cells rise super exponentially, giving you tons of inflammation and slowing regeneration, which determines all aging-related diseases, basically. And which one you get now depends on your genetics. But everyone has these rising senescent cells. So I believe that rising inflammation and reducing regeneration is what pushes the separatrix towards reduces the separatrix of the healing state. I believe that's right now. I believe that's a stochastic driver of aging, or major one. And if you're interested in that, you can read those papers. And of course, senolytic drugs that removes senescent cells can rejuvenate mice to a large extent. But there are other factors, so etc. David says, sure, TB growth hormone will protect the host from wider spreading of TB. You see this when you put patients on anti-TNF antibodies to treat arthritis. So I'll look into that. Whales have a lower chance of cancer, Marco. Correct, because they're so big. But I'm not sure about fibrosis in whales. And I think, for example, if the animal is prone to a lot of injury, or if there's a lot of parasites and bacteria in its environment, fibrosis is probably be easier, like better solution, because you want to seal things off. And then in the embryo has probably no chance to see a lot of pathogens. That's why healing doesn't go fibrotic. It's very regenerative. So I think those trade-offs will be interesting. Sydney, do regenerated cells after healing retain any immunological memory of its previous tissue architecture? I bet you they do. I bet you that their epigenetic state is altered. I don't know if that's true or not, but that would be my belief. And if anybody knows, please tell me. And will they be younger or older than the rest of the body is interesting, because maybe they rewash their altered epigenome. That's a fantastic question. I mean, oh, by the way, this reminds me of repetitive injury. I didn't tell you about repetitive injury. Suppose you have an injury that heals, and now I told you repetitive injuries, right? So you have an injury that heals. This injury heals, but here after a week you do a second small injury, boom, you're in the basin of retraction, you go to hot fibrosis. But if the second injury is after 3 weeks, boom, you heal. That's how repetitive injuries can cause fibrosis, even if each one is kind of small. But if maybe the tissue there is already epigenetically modified, so that's a complication. I love your questions. Vitaly, any memory of tissue architecture or immunological memory? Good question. I don't know. All right, I think it's a good time to finish. And I appreciate your questions very much. If you want to turn on your camera just to say bye-bye to me.
Thank you so much, Uri.
You can say bye. I really enjoyed.
Bye. Thank you very much.
Thanks. Great talk.
Thank you. Bye.
Thank you.
Bye,
Bye.
Jessica Fisher. Bye.
Mhm.
Yeah, thanks. Paula,
Yes.
was that understandable?
Yeah, I think so. There are a lot of interesting questions also, so that's
Yeah, lots of questions.
a good sign.
תודה, אורי, ביי.
Bye, Jasmine.
Bye.
Sungmin, do you want to say something? Ask me something?
Oh, not now. Yeah. Thank you very much for your great talk. Thank you.
Great.
Right.
Yeah, that was such a cool method of doing this.
Yeah, was it a part that was hard to follow?
I think no. For me, I haven't done this sort of topic, like the terms, the abbreviations, and these things that are quite technical. Maybe it was a bit hard to follow when you get into the mice experiments. But apart from that, I think it was generally fine.
I mean, the protein names and all this stuff.
Yeah, I guess for people who don't have a biological background. But I think the gist of it, obviously, like, yeah, it's very nicely explained and like stimulating with the questions.
Uri, may I ask you one question? I mean, this isn't a science question, but would you have any plan to give a talk in some conference? Because yeah, I'm looking for a conference to attend this year, but if you attend it, that
You'd
should
like
be
to
my
talk?
priority.
I am. I'm giving. Where do you live?
The Oxford.
In England? Mhm.
Yes.
I'm giving a talk in Milano on March 24th.
March, yeah. Very soon.
Very soon. And I don't have other conferences lined up. I mean, I'm going to give seminars in New York City,
I see.
March.
Okay. Yeah,
Thanks.
thanks for your
Very nice to hear.
Yeah, thank you.
Are you in Oxford, Paula? Cambridge.
I'm at Cambridge. Yeah.
Yeah.
So this is a seminar series based in Cambridge.
Okay.
Yeah.
Okay.
That's
Thanks very much.
cool. Thank you so much. Yeah. Yeah, great to meet you.
Great to meet you.
And
Bye.
hopefully see you at some. Bye.
Hopefully see you in
Bye.
person. Bye bye.